Hello students, welcome to the online tutorial for Vidyavan Higher Secondary School. Today this video is for class 12 science, and today I'll continue with the chapter Heredity and Variation. Let's not waste the time and let's get started with the chapter and let's see that how far we can complete the chapter. So now we will go to the chromosomal theory of inheritance. Okay, so what is the definition of this chromosome theory of inheritance? Here the definition says that Mendelian genes are located on chromosomes, and chromosomes as hereditary vehicles are segregated and assorted independently that takes place during transmission from one generation to the next. So what is being said over here, that the genes, where these genes are located, they are located in chromosomes, right? Now, this theory of inheritance, it is being said that chromosome and gene are being transmitted from one generation to another parallelly, together as a unit. Now, there are some evidences for this parallelism between gene and chromosome. There are 4 evidences. Number 1, evidences regarding what? Regarding that this gene and the chromosomes are transferred as an unit togetherly, okay, from one generation to the another. See, we cannot say that the chromosome are being transferred from one generation to another, but the genes are not transferring, because genes are located within the chromosome. And if these genes are transferring from one generation to another, so will this chromosome as well, because genes are located in this chromosome and chromosomes are containing the genes. So if these chromosomes are getting transferred, if these genes are getting transferred, so the chromosome have to transfer. Okay, so first one is that, first evidence that Mendelian factors and chromosome together are transmitted from one generation to the another, as I have told you just now. Number 2, the gametes, the haploid gametes contain the genes, and also these gametes contain haploid complement of homologous pair of chromosome. See what these gametes are containing. They are containing the genes. The gametes are containing genes, as well as they contain haploid set of chromosome. Haploid set of chromosome, that means 23 chromosome. Understood? Not 46 but 23. Number 3, that during fertilization, 2 gametes bearing haploid gene set along with one set of chromosome fuse to form diploid zygote. During fertilization, what happens? 2 gametes are there, each of n, right? That means they are carrying 23 and 23 chromosome, as well as they are carrying the genes. They actually fuse together and forms a diploid zygote, which is having 46 chromosome number. Okay. Number 4, as DNA synthesis takes place during S phase of interphase and synthesis of other components of genes and chromosomes takes place in interphase, parallelism between gene and chromosome is also evident. See, DNA synthesis occurs in S phase, as well as the synthesis of other proteinaceous components of genes and chromosomes also takes place in the S phase. So here also, from this point also, it is proved that yes, there is a parallelism between gene and chromosome. So this was all about the theory, chromosome theory of inheritance. Now next, we'll move on to the topic of non-disjunction. Now what is this topic, or what is it about? Non-disjunction means, that means it is the opposite of disjunction. Okay, disjunction means separate. Okay, so non-separate, non-disjunction, obviously. So what is it? It is a phenomena by which synapsed homologous pair of chromosomes, synapsed homologous pair of chromosome. What is this synapsed? What do you mean by synapsed? It comes, this what comes from the word synapsis. What is synapsis? During meiosis or mitosis, what happens? We have seen that these two chromosomes attached by the centromere, it is forming this type of structure, right? Right? So this structure or this combination of two chromosomes, these two are homologous chromosome, okay, connected by a single centromere. This process is termed as synapsis. That means two homologous chromosomes joined by this single centromere. This is called synapsis, and these homologous chromosomes who have taken part in the synapsis, they are termed as the synapsed homologous chromosome. Okay, now what happens in the anaphase, as you have read it in previous classes as well, that in the anaphase the homologous chromosome separate, okay, separate from each other, and they go or they move towards each poles. Like this, the homologous pair of chromosome actually separates in the anaphase. But what is this non-disjunction? Non-disjunction means that these homologous pair of chromosome fail to separate in the anaphase. That is why this process is named as non-disjunction. That means separation is failure in the anaphase. Now this non-disjunction is primarily of two types. One is mitotic, another one is meiotic. Now what is mitotic non-disjunction? Simply from your common sense, the non-disjunction takes place in the mitosis cell division. Now where this mitosis cell division actually takes place? In the body chromosome. So that means the non-disjunction which is taking place in the body chromosome or in the mitosis cell division. That is the mitotic non-disjunction. Another one is the meiotic non-disjunction. What is meiotic non-disjunction? The non-disjunction which is taking place in the meiosis cell division. That means during gamete formation, the meiosis cell division is actually taking place. So what happens over here? That the non-disjunction takes place in the meiosis or in the gamete formation, that is termed as the meiotic non-disjunction. Now this meiotic non-disjunction is again of two types. Okay, one is primary, another one is secondary. So primary meiotic non-disjunction, here the non-disjunction takes place in the phase of meiosis 1. See, the meiosis is actually completed in two steps, meiosis 1 and meiosis 2. Primary non-disjunction takes place in meiosis 1 and secondary takes place in meiosis 2. Got it? So this is all about the non-disjunction. Now there is another topic regarding non-disjunction, its significance. Now what is its importance or what is its effect, you can say. Now when as a result of non-disjunction, as I said, that the two chromosomes or two homologous chromosomes, they fail to separate. So as a result, if it is happening in meiosis or mitosis, whatever, so due to mitosis or meiosis, what is happening? One cell is divided into two, right? Now this is having 2n. Now this 2n should be equally distributed among these two cells, n and n. Now just think that the two homologous chromosomes could not separate. So what will happen? That due to this, one of the cell, resultant cell, will get one extra chromosome because they did not separate. It has gone with the chromosome, previous chromosome, and the other, as a result, there is an increase of the chromosome in one cell. So the other cell will face a lack of the chromosome. So thus creates the abnormality in the cells. Understood? Now the next is the chromosome. What is chromosome? Chromosome is the intranuclear fiber shaped or rod shaped, okay, gene bearing structure containing the DNA and some proteins, and it is also responsible for carrying hereditary units or hereditary messages from one generation to another is called chromosome. Chromosome, where it is situated? It is situated within the nucleus, right? So the chromosome, generally the body cells are having 2n set, or you can say diploid chromosomes, whereas the gametes, they are haploid in nature or containing n number of chromosomes. Now, what are the significance of chromosome? First of all, this chromosome is the hereditary unit. That is, it is carrying information, genetic information from one generation to another. Next is that with the help of crossing over, new variation or new genetic variation, new raw materials are provided for the evolution. Okay, number 3, that as it is the nuclear material, it can control all biochemical and physiological activities of the organisms. Along with that, number 4, that the stability of the genetic material is assured in chromosome. Okay, so these are the general significance of the chromosome. Now I will go to the structure. Now this is the general structure of the chromosome, as you can see over here. Generally the chromosome is made up of 2 threads or 2 fibers. Okay, now these 2 threads or fibers are termed as chromonema. Okay, now these chromonema actually coil around each other and forming a fiber-like structure, thus giving rise to the chromosome. Now how it is arranged? One of the coiling is like this. That means in the opposite direction. This coiling is termed as paranemic coil. Paranemic. Another one is, okay, this is termed as plectonemic coil. Okay, so these are the 2 types. Next, we will go that the chromosome is having 2 arms. Okay, and these 2 arms are being attached by the centromere. So this is the central centromere, and these two arms are called chromatids. Okay, these 2 arms are called chromatid. Now the short arm, suppose I think this is the short one. This short arm is called P arm and the long arm is called Q arm. Okay. Now the centromere, where it is located, it is having a constriction. Now this constriction is called primary constriction, which holds the centromere. Okay. Now the next is that these 2 chromatids are the sister chromatids of the chromosome, and these two ends of the chromosome is termed as the telomere. Okay, apart from primary constriction, the chromosome usually possesses another constriction. Okay, this constriction is called secondary constriction. Okay, now this constriction is actually associated with the nucleolus, and thus the secondary constriction is called nucleolus organizer region. Okay, next is that there is in the centromere there develops a fiber-like structure within the centromere, which is termed as the kinetochore. Okay, now what is this function of this kinetochore? This kinetochore actually helps in holding the spindle fiber during the cell division process. Next one is that at the end or at the tail end of the chromosome there is a satellite or a knob-like structure, and that is called the satellite, and the chromosomes which are bearing this satellite structure are termed as sat chromosome. So this was all about the structure of chromosome. Now coming to the next topic, that is the types of chromosome on the basis of number of centromere. First one is acentric. A means absence. That means the chromosome do not have a centromere, and this type of chromosome is not very stable. They generally get disorganized or destroyed. Next is monocentric, that means they are having one centromere. Next is dicentric, that means they are having two centromere, di. Next is polycentric, that means more than two. They are having more than two centromeres. Now the next is on the position of centromere. Metacentric, meta means middle, that means centromere is situated at the middle. Submetacentric means far away, a little bit away from the middle. Acrocentric means in between the middle and the terminal end. See, telocentric means at the terminal end. Next is that what is karyotype and ideogram. Karyotype is that the characteristics of the chromosome obtained from light microscopic view, its number, its size, its form, that is the karyotype. And ideogram is what? It is a diagrammatic representation of the chromosome. Okay. So I am keeping my video till this much. I hope you have understood. Thank you.